when we talk about this being a major topic for lecture exam 1, it's about how your body, your cells use these three to make ATP. Okay, does this help at all? And then the other major topic is DNA. And the two subtopics of DNA is how your DNA codes for protein synthesis, making proteins, and then also DNA replication. So on the whiteboard is over 85% of the points for lecture exam 1. Okay, so we ended up last time talking about catabolism and anabolism. Let's look at that, and then I'll put this on the whiteboard. Oops. And that was up here. Okay, so this is catabolism, and it says breaking down. Well, what that is doing, that is talking about, I really like that green one, breaking down chains and the links. This is catabolism in this direction for all four of them. If you break those things down, you're breaking, that's catabolism, and you're going to release energy. Which can be used for other things. And then the other one is just the opposite, and that's building up. And when you're building up, you're going in this direction. Oops, I better put this down one. Does this help understanding catabolism and anabolism, the building up and the tearing down with this concepts of links and chains? So here's catabolism here, here's anabolism here. And this is what it means on this one, on the whiteboard. But to build things up, and this has to do with the laws of thermodynamics, you have to put energy in. And this is the only form of energy your cells use. ATP. So if you're going to put energy in to build up any of those chains, you got to donate a lot of ATP. All right, now we got to get going. I will tell you right now, I'm setting up the concepts necessary to understand the first big topic, cellular metabolism. All this lecture today is setting up that topic of how your cells make ATP. What I tell students in this class, this is going to be your introduction to some things that are pretty intimidating. If you see a concept that is intimidating, resist the urge to be intimidated, because it does not help your hippocampus, which is in your limbic system. And if your limbic system is all messed up because you're intimidated, you don't remember shit. So you may as well not be intimidated, because if you're intimidated, you're going to flunk anyway. So don't get intimidated. Take a step back, see the big picture of what it's for, then break it down into sequential steps. And before you know it, because each step is easy to understand, the whole big picture is intimidating. Don't be intimidated. Take a step back and see what this big picture is for, then understand it in small, bite-sized sequential steps. And before you know it, I got this. You do that a few times, you're not intimidated in any classes anymore. If no teacher has ever made you do this, that's why it's intimidating. And the sooner you get over it and somebody teaches you how to do this, the sooner you can learn without being intimidated, okay? So what this lecture is talking about today is setting up this first big topic, and this is what it's for. Get this now, or forever be lost for the next hour. This is about how our cells, which only use this for energy, ATP, is going to use these links to make ATP. That's the big picture, bottom line. If you keep that in mind, you'll understand why you're learning the small bite-sized steps. You don't get that. You just, all the bite-sized steps are just swimming in your head. Does this help at all? Keep that in mind. All right. I left off on this slide on Monday. Protoplasm is basically the chemistry of the cell. It's made up, the top 5 things are water and ions. And then the 3 nutrients, which is why you got to eat them. Everybody got it? You have so much of those 3, that's why you have to eat them, so that you can get the links to make your own chains. All right, first we have to talk about carbon. Now look at what I drew on the whiteboard. I drew this, I think I drew it, yeah, last week. Does that look familiar with what's on the top right of this slide? Okay. Now, what makes this very special, because remember, you got to remember the 288 with electrons. What makes this very special is it makes the outer shell right in the middle of this 8. It's right in the middle of that 8, which puts it in a unique position. And this unique position causes it to chemically react with other atoms. Because of that, it is the topic of organic chemistry. If you are going to be taking organic chemistry, that has nothing to do with pesticides on produce. It has to do with the chemistry of carbon. Because of this, what I just put on the whiteboard, because the outer shell is right in the middle between 0 and 8. The electron that orbits the center of the atom actually doesn't orbit in circles. That's the oversimplification just for you guys so that you can understand how this works. It actually goes in these shaped orbitals here in blue. And that's why the bonds that it makes are symmetrical in 3 dimensions. When I drew it up on the whiteboard like this, it's in 2 dimensions. Carbon forms covalent bonds. That's electron sharing. Covalent bonds. It's going to form 4 of them all the time because the outer shell has 4 and it wants to get to 8. And those bonds are going to be symmetrical, which means these covalent bonds are extremely stable. It also has the lowest activation energy of all atoms. That probably doesn't make much sense to you. Just know that it bonds to anything, including itself. And it does so easily. In fact, out of the 85% of all known chemical compounds, 85% of them involve carbon. Organic chemistry is 85% of chemistry. Everybody got it? Because it's like this, it bonds to anything, including itself, so it's extremely versatile. If you want another analogy to help you understand carbon, it is the whore of all atoms. It'll hook up with anything, including itself. Okay? And because of that, carbon is the backbone to all these 4 biologically important macromolecules. Are there other atoms? Yes, but carbon is the main one. And so for your information for future classes that you're taking, organic chemistry is not the chemistry of life, although a lot of it is. It's the chemistry of carbon. Biochemistry is the chemistry of life. And to understand biochemistry, you got to understand organic chemistry. That's why It is in your education plans or your prerequisites. Organic chemistry is really important to understand. You cannot understand pharmacology until you understand biochemistry. You cannot understand biochemistry until you understand organic chemistry. Okay? Because our cells have these 4 biologically important macromolecules. Carbon is the backbone. We are a carbon-based life form, especially if you want to be a Trekkie. In fact, in organic chemistry, look at this figure. Organic chemists get tired of writing C's. So they write these figures instead. Each one of the corners of this hexagon is a carbon. Same thing here. Same thing here. This pentagon, all carbons. They just get sick and tired of writing C's. Everybody get how this atom is really important? All right. Now, the chemical reactions that we're going to talk about that help your cells turn these links into ATP are not random. These are chemical reactions are not random. They are specifically driven by enzymes. Okay? Now, you got to know what a catalyst is. A chemical catalyst stimulates faster chemical reactions without being permanently altered or used up in the process. That's what a catalyst is. And in chemistry, there can be various types of catalysts. In our cells, it is all done by enzymes. What you need to know is that enzymes are all proteins. I'm just going to put this underneath here as an example of proteins. All enzymes are proteins. One of the 4 biologically important macromolecules. It speeds up chemical reactions in our cells by lowering the activation energy of those chemical reactions. It's basically lowering the hump. So you have to understand that a chemical reaction starts with reactants, also referred to as substrates. These are terms you just need to know. The arrow is the chemical reaction, and then you end up with products. Okay, can everybody see that on the whiteboard? You start with chemicals that are reactants or substrates. It goes into the chemical reaction, and what comes out are the products. Everybody got it? Now, I'm going to run out of room on the whiteboard. You have to get that. By the way, for this lecture, people, be extremely clued in to everything I'm saying. Do not zone out for 5 minutes at anywhere. Because if you zone out, in 10 minutes, you're going to be lost. Stay focused. If you're not getting it, ask the question before you are lost. Because then you've lost 10 minutes of lecture, you're going to have a hard time catching up. Stay locked in all the way. Now, everybody understand these terms. Reactants are substrates that go into the reaction. Products come out. What you also need to know now, once you have that, is that it's not a straight line like I've drawn on the whiteboard. On an energy level, it's a hump. You have to put energy into this to do this reaction. And if you put enough energy into it, the energy activation, now it'll go through and the products will be the result. It's not a straight line. You got to go over an energy hump. Okay, I'm only talking about endergonic reactions right now. That's a whole nother chemistry thing. But these reactions, you got to put energy into it. Okay? what enzymes do is they lower the hump. And if you lower the hump, that reaction doesn't have as much of a barrier and it goes faster. So this is tied to this. That's how they work as biological catalysts, because they lower the hump. I'm just going to put it on the whiteboard. Hopefully that'll ring a bell when you look at it later. Okay. Without the catalyst, it's the blue line. With that enzyme, it's the red line. And it's going to go faster. It lowers the hump. From your textbook, it lowers the hump. Okay. Now, what's really important is you have to know that proteins are huge and they form like boulders, and like rocks have different shapes, so do proteins have different shapes. So since all enzymes are proteins, enzymes have different shapes. And this is what gives the enzyme specificity. It's three-dimensional shape, because on it are certain what are called active sites. Okay, and like on this animation here, the active site is that little triangle cut and the little round cut. Those are the active sites. If the right reactant enters the picture, it's going to speed up the reaction. But if another reactant comes in that doesn't have the right shape, it's absolutely inert to this substrate, to this reactant. Not going to fit? Nope, nothing. But if a substrate comes in that has the right shape to the active sites, like this, it will happen. It will lower the hump and boom. Now, what kind of reaction is this? Anabolic or catabolic?
Anabolic?
It's catabolic. It's breaking down one big block into two big blocks, into two links, for example. That would be catabolic. And then energy would be released. Okay, so the active site is where the reactant or substrate binds to the enzyme. And it binds, and we talked about this in the very first lecture, like a lock and key specificity. You can't put a square peg in a round hole. You guys remember that? That's the same thing with substrates and active sites. Got to be the right shape. Now, when the substrate does bind to the active sites, the enzyme may actually change, but it changes temporarily. And after the reaction, it snaps back into its original shape. This is why I said the enzyme is not permanently altered. It may be temporarily altered, but it's going to speed up reactions, and you still got the same enzyme. What kind of reaction is going on the bottom right?
Catabolic.
Yeah, that's also catabolic. That little blue line that you see entering the picture there, that is sometimes referred to as a coenzyme, which will also lower the activation energy and make it go even faster. Okay, so everybody understand this? I have it in the whiteboard in purple here for enzymes because it's really important. We're going to talk about a lot of sequential chemical reactions for your cells to make ADP. I'm talking about this because it is not, these reactions are not random. They are specific, and they are specifically driven by dozens and dozens of enzymes. Okay? And this is just showing you the same thing. Out of all the substrates, this enzyme is only going to work on this substrate. It's the only one that's going to fit. Now, all proteins, not just enzymes, all proteins are subject to what we call denaturation. Next to this slide, I would write lab 3. This is one of the reasons why I'm covering it now. And it's not going to be for a couple weeks because we started late with lab. But in lab 3, we will talk about denaturation. An enzyme or any protein, well, the enzyme has a range of conditions where it will work. We can talk about temperature. We can talk about pH. There's a range that it'll work called the range of tolerance. And it looks something like a bell-shaped curve. So look at these highlighted words here. I'm going to do it over here. Okay, so if this was, say, pH, from this point, it's 0. From that point, it's 0. But everything above there, it's above 0. This is the range of tolerance. Somewhere, usually in the middle, is the optimal. The optimal condition, or the optimal pH if it's not pH. All right? But if you are outside the range of tolerance, this is where you get denaturation. Both below this bell-shaped curve and above the bell-shaped curve. The conditions that are outside the range of tolerance. That's what I'm talking about right there. Okay? Does that help? So, we will play with this in lab 3. We're not going to do a group lab report. I should have a strike through that, but that's big in lab 3. What this is when you get outside the bell-shaped curve, you have a permanent alteration in protein structure. And I'll tell you a little bit later when we get to protein structure. But you change the shape permanently. Well, if active sites have a very specific shape and you permanently change it, it permanently destroys the enzyme's ability to work. Okay, there's that bell-shaped curve. The bell-shaped curve for enzymes are not always symmetrical, like how I drew there. And this is showing you a good example for the condition of temperature. And then we finally got kinetics. This has to do with reaction velocity. How fast is it going? Or reaction rates. Okay. There are things that will slow reaction rates or things that will speed it up, even with the enzyme. Okay. Do you want me to explain these underlying terms?
Yes, please.
Okay, well, obviously, if you have a higher concentration of enzymes, you're going to have greater, faster reaction rates. But when all the enzymes are working as fast as they can, now it's limited to substrate concentration. And remember, brackets means concentration of. So down here at the bottom of this left graph, it's substrate concentration. You go up in substrate concentration once you have a set amount of enzymes, and the higher the substrate concentration, the faster the reaction rate. That's velocity. Until you tap out. When it levels off horizontal, that means all the enzymes are working as fast as they can. You have just reached substrate saturation, where the line is horizontal. Again, I told you certain things can cause reaction rates to slow down. Those would be the red lines. The velocity is not nearly as steep then the black line, these will be the presence of inhibitors. But you can even get a more steep line here in blue if you have enzyme activation, and like that on that one animation, the little blue line. Coenzymes can oftentimes increase the velocity or reaction rate into the blue lines. Does that help? You can read about it in the textbook. It will not be nearly as simple as what I just described. Okay, I said up here on the whiteboard that we're looking at lecture packet pages 6 through 8. On page 6, it talks about the anabolism and catabolism we already talked about. It does talk about enzymes and the 3 major nutrient molecules we're going to look at. Page 7 is ATP. We already looked at that, but that's always good to refresh that. And then we're going to talk about some of the reactions that we see in this first big topic. Since we're talking about metabolism, how our cells make ATP out of nutrients, we better look at some classic metabolic reactions. Okay? Here we have the dehydration reaction, synonym condensation reaction. Here we see in this figure 3 links on one side, 1 link on the other side, but the 3 links has an H off one end. The single link has an OH off its end. It combines to form water, and boom, you got 3 plus 1 is now 4 link chain. Is this anabolic or catabolic?
Anabolic.
It's anabolic, written right here, because it's building up. You have 2 smaller molecules, you end up with 1 larger molecule that's building up, and you have to put energy into it. That's why it says highlighted, requires energy. This is why what I have over here on the whiteboard, that in anabolism, when you're building up chains, you got to put energy in. Okay? You got to put energy in to build up chains. The other side of that is exactly the opposite. And this is hydrolysis. Here you're inserting a water, and hydrolysis literally means breaking apart by water. You're using the water to break up this 4-link chain into 3 and a single link. So obviously this is a catabolic reaction, and when you do this, energy is now released. That is the orange arrow on the whiteboard. Everybody tracking with that?
Yeah, I have a quick question, Professor.
Oh,
Okay, in a catabolic reaction, the water separates. Doesn't it require energy to separate water into a hydrogen and a hydroxide?
No, because it is so much smaller compared to the bigger thing that is actually splitting apart. But this is splitting apart both of water and of the chain. Both are splitting apart. And when you break apart, catabolism, energy is released. Now look at both of these figures and both of these paragraphs and compare them. Is this not paired opposite content? Is this not paired opposite content?
Yes.
What are you supposed to do with paired opposite content?
Learn one.
Only one.
Learn one and learn the name of the other. Now you got synonyms for the first one. You got to learn both of those names. Dehydration which means water's coming off like that, or condensation, because it's like you got water out of nowhere. In these first two molecules, you don't have water. And all of a sudden, it's like water came out of nowhere. Kind of like if you park your car outside, you go out to your car in the morning, it's covered with water, condensation. The water was actually there, it was in the air. You just didn't see it on your car. Okay, but just learn one and learn the name of the other and know that it's opposite, and you won't get them switched. Okay, how are we doing on time? All right, let's look at the 3 types of nutrients now. The first 3 of the 4 biologically important macromolecules. Okay, first we have carbohydrates. And this is listed for you on the bottom of page 6. Actually, it's right in the middle. The name actually describes the molecule. There is a water for every carbon in the links. Once you start building up chains, it doesn't come out exactly that, but the single links mean that there's an H2O for every carbon. Okay? What you need to know is that the single links is the primary source of energy for cells. In other words, it's the primary fuel to make ATP. And that's why we're going to start with that when we do our discussion of this. Your cells will use this primarily. If it runs out of it, it'll use the other ones. But there is an H2O for every carbon. We see that formula in monosaccharides. Glucose is the most common in your cells. There's other ones that other plants make, so you have the body to use those as well. But it's basically you're going to convert it to glucose to use in your body, which is why blood glucose is an important lab test. But you also need to know disaccharides, and that means 2. So there's 2 links, because those are very common, like table sugar. Sucrose is a glucose and a fructose, and it's also very specific on how those 2 links are bound together. That's huge in specificity. But like if you eat a piece of bread which has starch in it, and that's a polysaccharide, but you don't make starch, plants make starch. You make glycogen, which is also a huge branch chain of hexagons. Look at that picture on the bottom. It's a huge branch chain. Each one of these little white things is a hexagon. Each one of those guys is a hexagon. Each one of those guys is a hexagon. You see how huge that thing can get? And that's both for both glycogen that we make and starch that plants make. But when you eat that piece of bread that has starch, your body will first break it down into disaccharides. That's the first digestive enzymes break it into disaccharides, and then the final digestive enzymes break it into individual links. And that's what your body absorbs, by the way. Your body can only absorb links in digestion. Okay. There it is there again, single links, boom, double links, and then the huge branch chain. This is actually incomplete. Glycogen would be much bigger than that. Any questions on carbohydrates? Primary source, primary energy source, or primary fuel your cells use to make ATP, and that's glucose. Let's go to lipids. Lipids are pretty diverse, but the thing about lipids is that they are hydrophobic because of fatty acids. Fatty acids are hydrophobic, non-polar. There's one exception. I'll talk about that in a bit. Okay, so remember I made this methane in week 1, and then I made this huge long hydrocarbon chain. Those right here. Huge long hydrocarbon chain. Now triglycerides I listed up here because this is probably the most common, outside of cholesterol, most common form of lipids in your body. And that's why if you look under the links, it says fatty acids, and then I put in parentheses, and glycerol. Glycerol is not a lipid, but it's this 3 carbon thing up on top. And if each one binds, well, what kind of reaction is this, by the way?
Anabolic?
It's anabolic, building up, but from those metabolic reactions, what is the specific name of that reaction?
Condensation.
Those are 3 condensation reactions yielding 3 waters. Everybody see that? But you got 3 fatty acids, you got a triglyceride, and they're all bound together on the glycerol. So that's what you see on the whiteboard way over there on the side. Under links, it says fatty acid and glycerol for this first bullet. The glycerol is not a lipid, but it's used to bind those together. What is the lipid that we saw in week 1? It's actually in the name of it.
Phospholipid.
Phospholipid. And it has 2 links. That's why it has 2 hydrophobic tails. Okay. So triglycerides have three fatty acids. Phospholipids have 2. But notice here that one of the tails of the phospholipid is bent. One is straight, one is bent. Everybody see that? Look at this bond right here, and we're going to get to that in just a second. This is what causes this one to bend. Triglyceride, again, the glycerol is in the brown box. Fatty acids are in the yellow boxes. Phospholipid, one of the tails is bent, and this is that same bond right here because of this. But this is the phosphate ion. This is a nitrate, a positive ion, and that's why this is polar. It's got a north pole, different from a south pole. The head is polar. But this is why that one tail is bent. The reason why that one tail is bent is because it has a double bond. And when you have a double bond, you'll lose 2 hydrogens. It also bends the chain. Up here, comparatively, both of these carbons right here have 2 hydrogens on each. Can you see that? You go down here to have a double bond, you only have 1 hydrogen. And the reason why is what I talked about over here on carbon. Carbon's going to form only 4 bonds. Let's look at this on the whiteboard here. It will always form 4 bonds. And so when I did the first thing last week on methane, I drew something like this. and I put dot, dot, dot. If it's actually a fatty acid, you're going to have this thing here called a hydroxyl group. And this is acidic because this H comes off as H plus, and that's why this is a fatty acid. But I drew something like this in week 1 when I was talking about hydrophobic and how this whole thing is actually considered symmetrical. Okay, everybody see that? Does that look familiar from week 1? Except for the carboxyl group on the end. Look at all those C's, all those carbons. How many black lines are coming out of all those C's?
4.
4. Even this guy. 1, 2, 3, 4. That is a double bond to oxygen. Oxygen will always form, if you remember on water, 2 covalent bonds. Here's 2. Here's 2. Hydrogen will always form 1. That's why out of each H, there's only 1 line. But carbon always has 4. Well, on that one figure, each C can only have 4 lines coming out of it. And now on these, for these 2 carbons, since there are 2 lines here, 3, 4, that's it. 1, 2, 3, 4, that's it. These 2 now have 2 less hydrogens. Can you see that on the whiteboard? And that's why this has as many hydrogens as you can have, with 4 bonds for every carbon. This is a saturated fatty acid. But because we have 2 less hydrogens here, this is unsaturated fatty acid. Now, does this slide make sense? In fact, what I drew on the whiteboard is a monounsaturated fatty acid. It has 1 double bond. If you have 2 or more, that's polyunsaturated fatty acid. And this has to do with dietary health. People are worried about cholesterol. Well, you shouldn't be worried about dietary cholesterol. Dietary cholesterol don't matter. What matters is that if you eat too much of this saturated fatty acid, your liver will make more cholesterol and you'll excrete less. If you eat more, especially polyunsaturated fatty acids, your liver will make less cholesterol and excrete more. But if there's cholesterol in food, that's not the point. The point is, what kind of fatty acids are you eating? And this is bad. This is good. It says that down here, underlined. Saturated fats have been linked to increased low-density lipoproteins, and that's the bad cholesterol, cardiovascular disease.
Which one did you say was mono and poly?
What I drew on the whiteboard is mono, unsaturated, because
Oh.
there's only 1 double bond. Can you see that?
Okay. Oh, yes. Okay.
If it has more than 1 double bond, then that's polyunsaturated. Okay, the rest of the lipids. Let's get moving here. Yeah, about a half hour. So this is a polyunsaturated, and you can see it has more than 1. But notice, for every double bond, you'll lose 2 hydrogens. Everybody see that? Okay. Ketones are also a lipid. It is also hydrophobic, but it is acidic. This is also, we'll talk about this later. Don't worry about this yet. I just want to just tie this later. This is a step product of beta oxidation. We will learn about that in next week, but it is a step. And so if you're doing a lot of this process called beta oxidation, you actually get acidic. And you can see here, we will learn about more of this in lab 8 when we talk about hormonal physiology and diabetes. Because people who are poorly controlled diabetics are doing a lot of beta oxidation. And that's why they struggle with ketoacidosis, because ketones are acidic. In fact, if you watch TV and there's a commercial comes on for, I don't know what the product is, Ozempic or something like that, and say, not for people with ketoacidosis. You have to be, you can't be out of control with a lot of these meds. Steroids. Steroids start out as a fatty acid. And then they form this classic 4-ring structure. And it is 6, 6, 6, 5. 3 of the rings are 6-sided hexagons. 1 is 5-sided. But since they start as a fatty acid, all of those are also hydrophobic. The one exception to hydrophobic lipids is prostaglandins. So that's why I put it on with a little blue asterisk. These are hydrophilic. The only lipids that are hydrophilic. And they do start out as a fatty acid. You can see it's a long chain fatty acid here, but it does a U-turn. And when it does a U-turn and forms one 5-member ring, now this thing has a north pole different from a south pole. Everybody see that? Because that pentagon is on one side. All of a sudden this becomes polar. Okay. Oh, by the way, all our steroids start as cholesterol. They all start as cholesterol. Then you just change a few active sites on the 4 rings and it changes it from testosterone to estrogen.
I'm sorry, I didn't catch that. They all start as cholesterol?
Yeah, all the different steroids in your body all start out as cholesterol.
Okay, thank you.
All right. All right. The third and last one is proteins. Again, the links are amino acids. This is the most diverse out of the 3 nutrients. This includes enzymes. It includes structural components of your body. In fact, when you look in the mirror, you're looking mostly at proteins. The shape of your body is skeletal muscle, actin and myosin, proteins. Connective tissue, collagen dominates connective tissue. But even on your skin, the stratum corneum is dominated by the protein keratin. So it's a major structural component, but remember, all enzymes are proteins. This is what I wrote it up here. Now what you're gonna have to be able to visually recognize, there will be some chemical structures on lecture exam 1, but it's gonna be simple, it's gonna be doable. I'm not gonna give you biochemistry. By the way, your textbook has a lot of biochemistry in it. And you don't need to know all that biochemistry. But when you look at a single amino acid, you need to recognize this, and you might want to make a note of it on the slide. The NCC. Okay. The NCC. This is the backbone of a single link amino acid. Nitrogen, where it is on the periodic table, that column only forms 3 bonds. Put this over here. Carbon is going to form 4, always 4. On the middle C is what is called the R group. And here's that carboxyl group again. Just like on the fatty acid. This is the amino group. This is the carboxyl group, which is always acidic because of this hydrogen here falling off. That's why it's called amino acid. Everybody got it? You have to visually recognize NCC. And when you do that, you can recognize the bonds between NCC. Okay, so I'm not going to draw the lines here. And that could be different amino acids. But the bond between links, amino acid links, are called peptide bonds, and you got to be able to visually recognize peptide bonds between NCCs. Okay. Everybody see that? And you can see in the green boxes here are the different R groups. But you need to know that out of the thousands of proteins in our bodies, there are only 20 amino acids, only 20 R groups. And you have the enzymes to make 12 of them. And here's that peptide bond between NCC. Everybody see that? Peptide bond is evidence of anabolism. And you have to do that dehydration reaction.
So for our exam, we'll have to recognize both amino acids and peptide bonds through images like this.
Yes, but you don't need to know the structures of the R groups.
So we'll just basically be looking if there's an NCCC in it, we know it's an amino acid.
Right.
And if there's something separating 2 NCCs, that makes it a peptide bond.
Right there.
Okay, perfect.
Yeah. Okay. Right. So you only can make 12 out of the 20. That means the other 8 you have to eat. Those are essential amino acids. You can't make them, you have got to eat them, or you're going to have a protein deficiency. Now, if you look at your essays, we have actually covered the last 4 essays on the second page. This next slide right here is the very first essay. Number 1, the 4 levels of protein structure. Everybody got your essay sheet out? This is the first one. Describe the 4 levels of protein structure. I'm not going to write this down because it's on the slide. Primary protein structure is just the amino acid sequence, the link sequence. That is formed by peptide bonds, like what you see on the whiteboard. That order 1, 2, and 3, that's formed by peptide bonds. That's primary protein structure. Then you keep adding links. And then you're going to start to get some hydrogen bonds between different links from different parts of the chain. These hydrogen bonds are weak, but they can form the chain into a helix, which was noticed first, so they called that an alpha helix. Or it can bend it back and forth like a pleated fan or a pleated sheet. And since they found that second, they called that the beta pleated sheet. That is secondary protein structure. You have to get a little longer chain to see these hydrogen bonds form. Hydrogen bonds are basically a slight positive being attracted to a slight negative because opposites attract. Does this make sense? This is why water, since it's polar, when you put water in a beaker, it actually forms a 3-dimensional network of hydrogen bonds. Okay? Tertiary structure is where the chain gets so big that other bonds will form, including hydrogen bonds, ionic bonds, disulfide bonds, van der Waals forces. Do you guys want me to explain van der Waals forces?
Yes, please.
Some R groups are hydrophilic, some R groups are hydrophobic. And like, in this case, it would be like, not opposites, but like R groups, hydrophilic and hydrophilic, have an attraction. Hydrophobic and hydrophilic have a repulsion. And that helps to bend the longer chain. But this is what gives a large protein boulder its specific 3-dimensional shape. And now I'm going to tie this to denaturation on the next slide. Well, after the textbook figure. We just mentioned it, but denaturation breaks hydrogen bonds. Right here, before I get any further, which structures do you see hydrogen bonds?
Secondary and
And?
tertiary.
That's right. So denaturation destroys secondary and tertiary protein structure because it breaks hydrogen bonds.
We can't see what you're writing on the whiteboard.
I know, you'll see later.
Okay.
Okay, so you have to get a pretty big boulder to be able to get tertiary protein structure, because the section that's alpha helix is going to bend to a section that's pleated sheet to another pleated sheet to another alpha helix, and it's forming this three-dimensional specific glob shape. And I use the word glob intentionally because that's actually used into some names of proteins like myoglobin, hemoglobin, because it has a specific glob shape. Okay. In fact, hemoglobin is one of the few examples we see quaternary protein structure, and this is where you get different chains, different globs arranged in a specific arrangement. In the case of hemoglobin, it's very symmetrical. But insulin, I told you, was a large protein because it also is a quaternary protein. These are the four. They pretty much all have the first one, unless it's really tiny, like antidiuretic hormone or oxytocin. That's all it has as primary. But you get, 20 amino acids, you're going to already start getting some hydrogen bonds, secondary protein structure. Most of the significant proteins have tertiary protein structure, and this is the specific three-dimensional glob shape. Only a few have quaternary protein structures, and you need multiple globs. This is from your textbook. Again, primary protein structure, peptide bonds, specific amino acid sequence. It gets longer, you start getting alpha helix, beta pleated sheets. That's formed by hydrogen bonds. Gets a little longer, you get specific three-dimensional glob shapes, different sections of pleated sheets, different sections of alpha helix. You need multiple different types of bonds there, but hydrogen bonds are also involved. And here's hemoglobin, 4 different globs. And that's an example of quaternary protein structure. Insulin has 2. But since it's 2 different globs, it's quaternary protein structure. Okay? And so this is that slide on denaturation. Larger protein globs are subject to denaturing, and that's the permanent destruction of hydrogen bonds, which affects secondary and tertiary right there. In lab 3, we'll mostly be talking about pH extremes and temperature extremes. But other things can cause denaturation as well, heavy metals, reducing agents, alcohol. But the best way to understand the permanence of this change is frying an egg. So you get the egg out of the fridge and it looks like this runny snot thing. You fry it in the pan and it becomes kind of more of a solid, rubbery thing. Well, that's because you gave it an extreme of temperature. That's what denatured the albumin in your egg. If you put it back, that egg back in the refrigerator, back within its range of tolerance, it doesn't go back to that snotty, slimy thing. It stays that fried egg. It's just cold. Denaturation is permanent. It does not go back. Okay, so use this fry an egg as an example that this is permanent. Go ahead and put that egg back in the fridge. It's not going to go back to that snot. Okay, so those were all the preliminary topics I wanted to talk about before actually getting into the first meaty topic for lecture exam 1. And this is how our cells turn the links of the 3 nutrients into putting that last phosphate group on ADP. That's how it's making ATP. It's not making it from scratch. It's just putting the last phosphate group on ADP. Okay? And we are going to, so just pay attention to this. This is really important. Remember, a monosaccharide is a carbon, a water molecule for every carbon. So the formula is going to be C6H12O6. That is a monosaccharide. We are going to start here. But this is all about putting the last phosphate group on ADP. How our cells put the last phosphate group on ADP. And that gets us ATP. We are going to start there. Once we have the framework of this, okay, because this is the big picture, is how our cells turn the 3 links into ATP. That's the big picture. Take a step back and see, why are we studying this? What is this all for? It is for that, because our cells only use that. Now we're going to break it into sequential bite-sized steps so you can understand it. If you can learn the sequence, this is extremely understandable. And you don't have to be intimidated. So there's glucose, C6H12O6. When we say the complete oxidation, I'll explain that more in a little bit. Maybe next week. Let's see how far we get. We need oxygen. And when we have oxygen, we're going to break up these 6 carbons into 6 CO2 molecules and water. This is the complete oxidation of glucose. And while we're doing that, we are putting 36 phosphate groups on 36 ADPs and getting 36 ATPs. This is the bottom line. Well, what we're starting with. We're going to start with this framework. Once I have this down, then I'm going to show you where these guys fit into this first framework. I got 10 minutes. We're going to cover preliminary principles. We're going to go cover the 3 steps. I'm going to break this into 3 sequential steps. And that's the 3 stages of cellular respiration. And if you want, and there's a formula up here, if you want to number them, here they are. Glycolysis is first. Then the Krebs cycle second. There is a tiny 1-step prep step that we're not going to call a 4th stage, all right? This is the 2nd step Krebs cycle. And then the 3rd step is the electron transport system. Notice in our yield of 36 ATPs where the big payoff is. It's in the last step. You only get 2 in the first, you only get 2 in the second, well, you get 32 in the last. That's the payoff.
Can you
Now,
go back to the beginning of the last?
This one?
One more back. Right there. Thank you.
Okay. Again, your textbook has a lot of biochemistry in it. Concentrate on the summary figures and the summary portions of this. This is not a biochemistry class. Okay, all right. We'll go over this preliminary principle here. Oh boy, I gotta get going. First of all, we're going to have an exchange of high energy electrons. And this exchange of high energy electrons, and this is really important, especially if we're talking about the last step here, the electron transport system. We're talking about high energy electrons, okay? And if you're going to understand the 3rd step, where there's the big payoff, you got to understand this preliminary concept. This exchange or transfer of high energy electrons is called oxidation reduction reactions. And they are always coupled, which means you always have one with the other. You never have one by itself. If you have reduction, you're going to have oxidation. And this is where the term complete oxidation of glucose comes from. So if there's high energy electrons here and compound A is going to give it to compound B is said to be reduced because it gained the high energy electrons. Now that might seem contradictory. If it gains it, how is it reduced? Well, maybe remember that electrons are negatively charged. And if you gain something negative, you're reduced. Will that help? And A, which lost electrons, is considered oxidized. And the reason why it's called oxidized, because in this sequence, the last electron acceptor is oxygen. It's the final electron acceptor. So it's the final oxidizing agent. So I say, well, if it's oxygen is the final agent, we're going to call it oxidizing agent. If you want a simple mnemonic to remember it, remember Leo Grr, as in Leo the lion says grr. Lose electron, oxidation, gain electron, reduction. Now the way a lot of this reduction is going, especially in the first two steps, are with these electron carriers. And electron carriers are also dinucleotides. So NAD and FAD have two nucleotides, dinucleotides. Now what you want to see here with this is remember that hydrogen, when we saw this with water, hydrogen is just an electron and a proton. That's all hydrogen is, electron and a proton. These high energy electrons are not free electrons. And that's what it says here. The electron, the high energy electron, is in the form of a hydrogen atom, a proton and an electron. All right, so off to the side of this slide, you should write, if you see H's, that's the high energy reduced form. What I wrote on the whiteboard there in purple, you don't see H's. That's the low energy oxidized form. If you see H's, it's got the electron. If you don't see H's, it doesn't have the electron. So here it is right here. This is the low energy oxidized state in yellow. This is the high energy reduced state in white. Same thing here. And actually NAD, it looks like it only gains one, it actually does gain two. What's sometimes referred to as NADH plus H, because the H is actually not bound to it here in red. So you'll see NADH plus H and NADH2. Oops. Since they can swing from both sides of the plate, they could be either reducing or oxidizing agents. That's why they're electron carriers. They go up here where there's a reaction, they gain high energy electrons, and they take it to the third step. They drop it off, they come back and get more high energy electrons. They go back to the third step, drop them off. They don't hang on to them, they're just electron carriers. Okay. Now, of those 6 carbons, we are eventually going to break them all off into CO2s, and we're going to do it one at a time. 6 times, we're going to get 6 CO2s, and every time that is a decarboxylation reaction, you're losing a carbon, that is actually catabolism. So this catabolism here is what's happening in the GI tract. This catabolism here in the 6 CO2s, that's in cells. But both of it is catabolism. This catabolism from chains to links is done by your digestive tract. This catabolism from 6-carbon glucose to 6 CO2s, that's happening in cells. And that's what we're talking about here, is what's going on in cells. And to do that, 6 times, you got to do a decarboxylation reaction. You release energy, and where does that energy go? Look at this. Right here. Low energy oxidized state. Now it's the high energy reduced state. That's where the energy went. Where did the energy comes from? Catabolism. Energy released. When you took that carbon off. Also, there is phosphorylation, and there's 2 different forms of phosphorylation here. And they're highlighted down there, substrate level or oxidative. Ooh, I gotta get going. What you want to write next to number 3 here, down at the bottom, substrate is where the carbons are. If you make ATP where the carbons are, that is phosphate level phosphorylation. If there are no carbons there, that's oxidative phosphorylation. Look at this figure again. Glycolysis, carbons are still here. This is substrate level phosphorylation. Krebs cycle, the last carbons come off as CO2 here, but this ATP is still where the carbons are, substrate level phosphorylation. And look what your textbook calls the electron transport chain. They don't even call it the electron transport system. They call it oxidative phosphorylation. ATPs, there's no carbon. Okay? Since we're starting with glucose, these are the 3 main sources of glucose for your cells. We already learned about facilitated diffusion, very simple. Then number 2 is glycogenolysis. Remember that glycogen is the carbohydrate, huge branch chains. If you break off glucose links off of this huge branch chain, you are breaking apart glycogen, glycogenolysis. That's another source. Okay. And then finally, I need to teach you this word. This is new glucose generated from nothing. What is Genesis? Genesis in the Bible is God created everything out of nothing. What this literally means is it's making new glucose, new glucose from non-carbohydrate sources. So these are not carbohydrates, and your liver can actually make glucose out of them. That would be gluconeogenesis. So the 3 sources: facilitated diffusion, glycogenolysis, splitting apart glycogen, breaking apart links, or making new glucose from other molecules, gluconeogenesis. Okay? There's various pathways, and your liver does all of that. And here are the 3 steps in order. We're going to start on this on Monday. Glycolysis, we saw that in that figure. You could see it in that figure that I went back to. Glycolysis, Krebs cycle, electron transport system, with the big payoff being in the 3rd step. Okay, we did it. Any questions? Let me show you the whiteboard.
Question.
Go ahead.
Not really reading the lecture, but I just wanted to ask if we're still gonna have the practice quiz type of thing.
Now, if we do that, we got to do it next week because I think the following week is when we have our first lab quiz. But you will be getting actually a long Canvas announcement on Friday that is showing you the protocols of proctoring by camera, webcam. I will, hopefully I have time. I mean, I'm going over on every lecture because I'm taking the time to make sure you guys get this. I might have to, if I'm going to cover that and not just depend on you to read it, I might have to go a little faster in the lecture. But it's coming Friday, definitely read it. And then if you guys want to do a practice lab quiz in lab, we could set something up next week. Okay, so this is the left bank showing you the 4 biologically important macromolecules with the links and the chains, catabolism, anabolism. All enzymes are proteins. Proteins are formed by peptide bonds. Metabolism that we're going to really get into next week is we're going to start with monosaccharides, glucose, and then we'll build the other nutrients off that framework. And then there's carbon, enzymes with a little graph about denaturation, and it breaks hydrogen bonds. Since it breaks hydrogen bonds, it permanently destroys secondary and tertiary protein structure. And then the 3 nutrients. Carbohydrates and lipids. I drew out for you the difference between saturated and unsaturated fats, and then there's the 3rd nutrients on proteins. Any questions? All right, I'll see Wednesday lab. Gosh, in about 24 minutes.